All right, hello IB biology students. This is Miss Sheely again. Today's notes are still in chapter 3, our genetics chapter, and this is section 3.5, genetic modification and biotechnology. The first thing we're going to talk about is something called polymerase chain reaction. Now, most everything in this set of notes you have actually learned in baby bio, so this should just be a review or maybe taking it to the next level. So remember that a polymerase chain reaction, well, our polymerase, we've talked about RNA polymerase and DNA polymerase. Well, a polymerase chain reaction is used to amplify small samples of DNA. So for amplifying samples of DNA, we're making more copies. So this is going to be a use of our DNA polymerase enzyme. This is really important or helpful in order to use those samples for DNA profiling, for recombination, to be able to identify species, and many other uses in research. So the process needs a few things. We need something called a thermal cycler. We need primers. We need free DNA nucleotides, and we need the enzyme DNA polymerase. The first thing that we do is we heat the DNA, the original sample, in the thermocycler, and we do that because the heating denatures the hydrogen bonds and exposes the bases. Then we cool the mixture, and when we cool the mixture, we then add primers to the start of the target area of the genes. So we don't necessarily want to copy the whole thing. If we're going to use it for DNA profiling, then we just want a small section of the DNA in a particular genes that we can use to compare to other samples of DNA. And then we allow the DNA polymerase to replicate the DNA using just our normal complementary base pairing. DNA polymerase does its job just as if this was normal replication. But then we repeat this cycle many, many times until we get thousands of copies of the DNA. And this is actually enough to amplify even a tiny sample found at a crime scene. So those two videos are just two different ways of looking at the same thing. So what you're trying to get is just a target sequence. You could really see that target sequence very well in that last video. All right, so our next concept is the idea of DNA profiling. DNA profiling is just comparing the sections of DNA between individuals in order to determine paternity or relationships, as evidence in crime scenes, or even to identify a species. So we use a process of gel electrophoresis, and we're able to examine the fragments of DNA after they're moved through an electric field and separated based on their charge. So how does this happen? Well, first of all, we have to take the DNA samples and then we amplify them with PCR. We want to make sure that we have a lot of samples of the DNA and we definitely don't destroy our original sample, especially if it is a criminal case or maybe even a new species. So then we're going to use something called a restriction enzyme, and a restriction enzyme is an enzyme that cuts DNA fragments, and it cuts them at specific base sequences. So depending on the target sequence that we're looking for, that's going to depend on which restriction enzyme we want to use. Then we're going to use a fluorescent marker to bind to a triplet in the DNA fragments, and that's going to allow the results of the DNA to be seen. The samples are then added to a gel electrophoresis chamber. The electric current is then hooked up and turned on. And when the electric current is turned on, it pushes the fragments from the negative end of the charge to the positive end of the current. Heavier fragments are going to stay closer to the original well, and smaller fragments will go further to the end of the gel. And when everything's done, you can stain it and you'll be able to see a banding pattern for each DNA sample, and then those samples can be compared. So this is a lab that we will do in class so that you can really see the hands-on kind of how this works. It's a really fun lab. All right, so DNA profiling, continuing with the gel electrophoresis. So we take a sample of DNA. We're going to use that PCR that we talked about to get a useful amount of DNA. We're going to use the restriction enzymes to cut the DNA. We're going to put it into a gel electrophoresis. So we've already talked about all of this. But this is this gel here is what it would look like when it is finished. So in this case, you have a marker or standard. So this is just a typical DNA ladder that you can use as comparison, okay? And then you have, here's your wells where your original DNAs were placed before the electric current was turned on. So this end would have been closer to the negative, and then this end is closer to the positive. So these pieces down here are much smaller than these pieces up here. There's a much larger. All right, so when we're looking at DNA evidence to identify suspects, we can use it to actually convict a suspect, or we can use it to eliminate someone as an innocent from an investigation. So in this case, we have a hair follicle that was left at the scene of a crime. We need to figure out who the perpetrator was. So our first column is just our standard. Our column A is trace evidence. That was the hair follicle. We have column B, which is the homeowner. And then we have two suspects, suspect 1 and suspect 2. When we look at this, we want to look at the trace evidence. When we look at the trace evidence, we are looking for matches in the other columns. So as you can see here, the homeowner, the band does not correlate in either of these two bands, so the homeowner was definitely not the perpetrator. We can eliminate them as an innocent person. Suspect 1 does match up both here and here, so that does indicate that he is probably the suspect. He does match up 100%, and you can confirm that suspect 2 does not have any matches. So suspect 1 is the perpetrator, as he has a 100% match with the hair follicle. When we use DNA profiling in paternity cases, it's a little different. Now we're looking at a relationship between the child and the parent, but you have to remember that a child gets half of its genetic information from its mom and half of its genetic information from its dad. So it's not going to be 100% matched like it is in a crime scene. So in this case, we still have our standard. Column A is going to be the mom. Column B is the child in question. And then we have two possible fathers. So the first thing we want to do is highlight our child. So here is our child and who we're trying to figure out. And then we're going to look and see if there's any comparisons or similarities with mom. And we want to take those into consideration. So if you look, there are two band placements that are similar to mom. So then we want to compare and look at where these open bands are that are not in mom and who they compare to. So we highlight them across, and the first band is only in man 1, column C. Looking at the second open band, he is also in man 1 or column C. So the father of this child is man 1. But if you look, there are some similarities between C and D. So these indications, these similarities actually indicate that man 1 and man 2 may actually have a familial relationship. They might be brothers, or they might be father-son, that type of relationship. They are related in some way, more than likely. All right, so some other uses of genetic engineering. Genetic engineering is also known as genetic modification. It can be known as gene transfer, and it can also be known as transgenics, or the field of transgenics. So remember that all living things all use the same bases. We all have A's and G's and C's and T's and U's. We all use the same genetic code. And each codon produces the same 20 amino acids in transcription and translation. And that doesn't matter. It is regardless of the species. So it doesn't matter what the species is. We only have those 20 amino acids. So with that idea, we can take into consideration that the sequence of amino acids in a polypeptide is going to remain unchanged. Therefore, we can take genes from one species and we can actually insert them into the genome of another species. And we do this by taking a bacteria cell, and we're going to use the plasmid, or their small circular DNA. We're going to cut that plasmid open. And then we're going to take a piece of DNA. In this case, it's a piece of the gene, or it is the gene that codes for human insulin. And we're going to insert it into the bacteria. And when that bacteria, that plasmid with the human insulin gene, is inserted back into a new bacteria, the bacteria will divide, and that bacteria will actually begin to produce insulin, even though it doesn't need insulin. This is actually how we manufacture chemical insulin. So we can use this idea of genetic engineering and that the genetic code is universal among species to have another organism do the work that we cannot. All right, so let's look at this idea of gene transfer. Again, it's kind of just what I went through in that very, very small little animation, but I'm going to add a few more details to it. So gene transfer, like I just said, requires plasmids. It requires a host cell, restriction enzymes, and an enzyme called ligase. Now, we've not talked about ligase before. So we're going to use those restriction enzymes to cut the desired gene from the genome. For example, the gene that produces insulin in our bodies, in human bodies. And then we're going to take a bacteria. The bacteria that's used is typically E. coli, and we're going to make sure that that E. coli bacteria has the small circles of DNA called plasmids, and we're going to remove those. We're going to use the same restriction enzyme to cut the plasmid in one place, and to cut the restriction enzyme, and to cut the gene in our human DNA, and that's going to cut in two places so that we get a small section. Now, restriction enzyme cuts in a Z cut, okay? So in this particular example, it cuts between the G and the T here and the C and the T over here. And what we get are these nitrogen bases that are left unbonded. And these kind of, because they're exposed and they're able to form hydrogen bonds with other nitrogen bases, they're said to be sticky, and they can stick to other sticky ends. So we'll put that gene into the plasmid, like so, and those sticky ends will touch, and then we'll use an enzyme called ligase. The enzyme ligase kind of works like a glue stick, and it just ensures that those hydrogen bonds are formed between those nitrogen bases that were originally left exposed. So ligase is going to join the sticky ends, and it's going to make that a complete, much larger plasmid. This new DNA is now called recombinant. Okay, so it's recombinant DNA because it's a plasmid. It's a recombinant plasmid. We now take that recombinant plasmid and we're going to insert it into our host cell, in this case, our bacteria. And that is now going to be able to express our insulin, be able to express the gene and make our insulin. All right, so we've used this concept before of recombinant DNA and recombinant genes to make something called a genetically modified organisms. GMOs are already in circulation. They've been produced for many uses, and many, including agricultural and medicinal. So we've used the medical, that was our previous example with insulin. But some of our plant examples, we've actually made something called golden rice. Golden rice is enriched with beta carotene. Beta carotene is converted to vitamin A in the body, and this actually helps prevent malnutrition related blindness in developing countries where they have plenty of rice, but they don't have a lot of sources of beta carotene. We've also been able to make insect-resistant corn. This is typically called BT corn. This was first produced by a company called Monsanto, and this produces proteins which the pests don't like. Therefore, toxic insecticides are not needed on the farm. And then we've also been able to make salt-resistant tomatoes. This allows tomatoes to be grown in saline soils or high salt soils. There are many, many other plant examples. We have strawberries that have genes from Atlantic cod that are able to survive frost, and just many, many other examples. It's kind of fascinating if you do some research on it. Some of our animal examples, we've been able to have factor 9 producing sheep. So these sheep produce the human clotting factors in their milk, and then that is helpful for patients or people with hemophilia, so they can be used to treat hemophilia. We also have glowing pigs. Cells from these specimens are used to study transplants and grafts and the final destinations of transplanted cells in the host body. So by having these transplanted parts like pig hearts and having their cells be able to glow, we can be able to really study the effect of a transplant and the graft and how those cells end up working in the host body. But as you can probably starting to think, there's a lot of discussion about genetically modified crops and specifically crops, and not necessarily in the human uses. But some of the benefits, we get a huge increased yield of crop, and we get faster breeding cycles of organisms. We get crops that can be grown in harsher environmental conditions. We have a reduced need of pesticides, which can harm human and environmental health through biomagnification. We'll get into biomagnification a little bit more in our next unit, but it's just a buildup of toxins in a food chain. Nutrient enhanced crops like the rice that we just talked about are helpful in areas of high food pressure or famine. With any benefit, there are some harms. So there is potential genetic pollution of organic crops through fertilization of the pollen of genetically modified or GM crops. There's unknown health risks. We don't know what it means for us to eat this food, and if it has the genes from other organisms, is it safe to eat? For example, the BT corn. We don't eat anything else that produces these toxins that the insects don't like, so is that even healthy for us? There is some fear of monopoly-like behavior as farmers need to buy these expensive seeds annually. They can't, there is a fear of monopoly-like behavior as farmers need to buy expensive seeds annually. So they would need to buy the seeds for that BT corn every year in order to ensure that they are not having to use as many pesticides. And then we also have the potential for hybridization of related species. So could a BT corn fertilize a regular corn species, or vice versa? So, and how would that affect? All right, our third topic today is cloning. Clone is a group of genetically identical organisms. A clone can also be defined as a group of cells that are derived from a single parent cell. So we have just twins. These are your natural identical twins. They're naturally occurring clones. So how can you tell the difference between twins? If you have known a set of twins for a while, at first you may not be able to tell them apart, but over time you can. How is that possible if they are naturally occurring clones? Also, plant cuttings. Plant cuttings are an example of clones. You take a small piece of a plant and you're able to grow a much larger plant. How is that even possible? Asexual reproduction is also an example of a natural type of cloning. So that's binary fission in bacteria. You take one bacteria, and through binary fission, you're able to make two bacteria that are exactly identical to each other. So all three of these examples are examples of natural cloning processes, or cloning processes in nature. But we also have man-made cloning. That's typically what people think when they think of cloning. The first type of cloning, and this is what you most commonly think of, is called reproductive cloning. It's pretty simple. You take a differentiated diploid nucleus from the individual that could be cloned, a skin cell. And then you take a donor egg cell and you enucleate it, which means to remove the nucleus. You take the diploid nucleus and you put that into the egg that has no nucleus. And then you implant that whole thing into a surrogate, and you allow that cell, that zygote, to go through gestation. And the newborn will be genetically identical to the nucleus of the parent. If you might remember from Baby Bio, most of our classes do this click and clone. This is from Learn Genetics. You can go through it again on your own. It's a pretty nice little tutorial there. clone some mice. Dolly the sheep was the first successful cloning of a mammal from a differentiated somatic cell. She was the result of many attempts. It did not happen on the first try, but interestingly enough, she died young, but of age-related illness. So we're not. And of course, human reproductive cloning is illegal. So the other type of cloning that we have is called therapeutic cloning. Therapeutic cloning is very similar to the to reproductive cloning. We still take a differentiated diploid nucleus from a cell that we want to be cloned. So this would be a particular cell, maybe still a skin cell. We're going to take an egg cell that's been enucleated where the nucleus is removed, but we have the rest of the cell. We're going to insert the nucleus into the egg cell. So far, that's exactly the same. We're going to stimulate this to divide and grow, but this is going to be done in vitro in a laboratory. And we get an embryo at that point. The resulting embryo is a rich source of stem cells, and those stem cells can then be harvested or cultured. So the way that we do that is we remove the outer area of the outer layer of cells, and so that we just get this inner mass, and this inner mass is the source of stem cells, which we want, and we can use that to culture the tissues that we need. So what would we use therapeutic cloning for? We're not going to make an entire organism. In this case, we're going to make stem cells for transplants, such as in burn patients, so skin transplants, or even leukemia patients. We can use it to replace other damaged tissues, such as nerve cells or nerves themselves, pancreatic cells. We can also reduce the risk of injection of cells that are genetically identical to the recipient. So the last use of a therapeutic cloning is to reduce the risk of rejection of cells that because they are genetically identical to the recipient. So just like with cloning, there's some, just like with genetic engineering and the GMOs, genetically modified organisms and food, we have ethics of therapeutic cloning. So this is a hot, contentious debate. Your arguments in favor of therapeutic cloning: stem cells can be created without the need for fertilization and destruction of natural human embryos. They're a source of cells for stem cell transplants, such as in leukemia, diabetes, burns, and other medical cases. Transplants don't require the death of another human being. Someone doesn't have to die for that person to get a transplant. Transplants are less likely to be rejected as they are cells which are genetically identical to the patient. So there's less likely of failure or rejection. And embryos are not allowed to develop to the point where a nervous system forms, so there's no pain or perception. But of course, we have our arguments against. We have a religious or moral objections due to the idea of playing God. The embryo which is created could potentially be used in IVF or in vitro fertilization and thus develop into a human fetus. So at what point are we considering that we're creating life just to destroy it, even though the nervous system has not formed yet? And then although cloning humans reproductively is illegal, this has not been ratified by all nations, so there is potential for a race to clone for the first human. That's the end for today's notes. I hope you found this as a great review from 9th grade baby bio, and I hope you find that the labs we do are super fun. Thanks so much.